So the goal of this course is to give an introduction to functional brain imaging approaches. And so what is functional neuroimaging or functional brain imaging? It's any technique that allows you to examine dynamic or changing cerebral processes during resting states or sensory or motor responses or cognitive challenges. So memory tasks or motor tasks where you're tapping your fingers or visual stimulation. And so it allows us to look at how is the brain changing, not just a static picture, but how is it changing over time? So there's a range of techniques, and they all have different degrees of invasiveness, but most are non-invasive. That is, they don't enter the body. So anytime we refer to, is a procedure invasive, is it harmful or painful? And only one of the techniques we'll discuss requires injection of a substance, and that is PET. We'll talk about that shortly. But ultimately, the goal is to detect signals from the surface of the body. And there's different degrees of spatial and temporal resolution, two important concepts. So with regard to resolution, spatial resolution is our ability to discriminate in space. So any details we can see in space, how close to the neural event are we? To the best spatial resolution would be the level of the neuron. But temporal resolution is a different dimension, and that is our ability to understand when an event happens in time. So how close to the timing of a neuron firing is our measurement. And there's typically a trade-off between these dimensions. As spatial resolution increases, you often lose temporal resolution with your technique, and vice versa. As temporal resolution increases, you often lose the ability to discriminate where something is happening in space. So this is your cheat sheet for that effect. This axis here, the y-axis, is in millimeters. This is space. And the x-axis is in time, seconds, from milliseconds all the way up to about a second, right? Or longer. And spatial resolution, you'll see EEG, which we'll talk about in a moment, and magnetoencephalography have really fantastic temporal resolution. But EEG has less specificity when it comes to space. So on the order of 20 millimeters or 2 centimeters, right, which is very large area in the brain. fMRI, which we'll also talk about, functional magnetic resonance imaging, has very good spatial resolution on the order of millimeters. Now, again, this is not very specific like you would have in animals, but for humans, that's pretty good, 2 millimeters or so, or maybe a millimeter. But the temporal resolution, you give up a little bit. It's on the order of probably around 2 seconds. So when we talk about imaging the brain and viewing the brain, there's different views that you should be familiar with. And the first is sagittal, and that's when we look at slices of the brain or images of the brain. We're looking at the left or the right. Imagine looking in by the ear, left or right. And then coronal would be if you look at the face of somebody, if you remove their face and their skull, you're looking straight at the brain from the front or the back. And axial would be if you get up on a ladder and you look down from the top, down on somebody's brain. Technically, I suppose it could be from the bottom. Usually we think of imaging from the top, and then transverse and horizontal are other terms for an axial view. So there are different families of functional brain imaging methods. And the first we'll talk about are PET and SPECT. They're both techniques that are based on blood flow or perfusion, or looking at how large molecules that are metabolized in the brain. These are commonly used in hospital systems, but they are also used in research. So PET, or positron emission tomography, is an example of a technique that examines brain function based on cerebral metabolism. In order to do this, you do have to inject an individual with a compound that breaks down. That's called a radioactive isotope, but it doesn't include radiation per se. And over time, it's been established that PET is safe for individuals, as long as it's not repeated. This is not something you would do daily, for example, but What you're really trying to do is, after you inject this compound, you look at how it breaks down in the system, and so how it is metabolized. And you can use different compounds to look at how, for example, oxygen is being metabolized, or glucose. And those would be two areas. If it's high oxygen utilization or glucose utilization, that means there's high areas of neurofiring. But PET is also used to look at abnormal aggregation of proteins, like in Alzheimer's disease. And so this is a PET CT system, actually. And so the person lies down on the bed, and their head would go back into the bore, or this opening, and they're injected with a tracer, and then they can examine how that compound decays over time, and it decays rather rapidly. And so this is an example slide of O15, which is an isotope for water. And it shows how much oxygen, because water is H2O, how much oxygen is being utilized in the brain. And if you look here, the red and black shows higher oxygen metabolism that's being used. And then the areas of yellow are the ventricles. It's just cerebrospinal fluid where there's no neurons, and therefore there should be no oxygen being metabolized. In this case, this is fluorodeoxyglucose, or FDG PET. And so it tells us how much glucose, sugar, is being metabolized in the brain. And this is going to increase in areas where there's more neural firing. And so if you look at the scale, where red is high glucose metabolism, and blue, purple, and black is lower, again, we see the ventricles are purple or blue. There's no neurons firing, and the red is higher. And so there's more neurons firing in these areas. And you can look at this would be comparing A to B, where A is an individual who does not have Alzheimer's disease, and B is somebody with a neurodegenerative process going on. And we see reduced metabolism throughout the brain. So there's less neuronal firing. A different family of functional brain imaging methods are electrophysiological methods. And so unlike the cerebral blood flow perfusion techniques, where we're looking at the breakdown or decay of a compound to tell us how much blood was flowing, or how much metabolism was going on in an area as an indicator of neural firing, electrophysiological techniques have direct measurement of neural firing. What they lack commonly is spatial resolution. So they're fantastic temporal resolution in the sense that this is EEG. These are electroencephalogram or electroencephalography caps. And each of these electrodes gives us a direct measurement of neural firing over time, which is really fantastic in the sense that the temporal resolution is on the order of milliseconds. The spatial resolution is not as good as MRI, which we'll get to in a moment. But when you wear a cap like this, now we can have signals from each of these electrodes, and we can begin creating a map of electrical activity in the brain. And this is an old technique. The original EEG is almost 100 years ago by Hans Berger. And in essence, what was demonstrated then is if you average signals and you had just a couple of EEG electrodes, you could get signaling from the brain, even if it's all averaged just as a single signal, which still had fantastic temporal resolution in time on the order of milliseconds. But the spatial resolution, if you have a single signal from the entire brain, which is roughly 80 billion neurons, the spatial resolution obviously is lacking. In EEG, you can also break down the different waveforms. And what you're looking at is the shape, or the magnitude, the height of that waveform, but also its frequency, how often you see it. And so, for example, a delta sleep, which is a slow wave, or delta associated with sleep, that's a slow wave. delta. Theta is a higher frequency, occurs more often, and it's associated with cognition. And all these signals are detected from the scalp. They're amplified roughly 1 million times in order to see them. And so you look at, again, amplitude of the shape and how often you see that signal, the frequency. Magnetoencephalography is a related technique, although it uses very different technology. It uses, this looks like almost like a big hairstyling machine. And in essence, what it's doing is picking up magnetic flux that emanates outside your head when there's neural firing, which sounds like science fiction. It sounds like something you'd see in a movie. It's completely non-invasive, meaning you don't have to inject anybody with anything. It sits just outside the head. It's not hazardous. You can repeat it. You can sit in the MEG machine for time and it doesn't hurt you. And it measures the brain's electrophysiological events at about 1 millisecond, which is fantastic, much like EEG. What it also gives you, though, is better spatial resolution. And so what you're picking up is this electromagnetic energy. And I'll show you a better picture of what that looks like in just a moment. But it travels outside your head where it can be recorded. And so you have these magnetometers are based on superconducting loops. It's positioned just outside the head in order to detect these tiny, tiny signals. So this is the magnetic flux. Anytime we're thinking about things, memories, daydreaming, or active mental processing of math or reading a book, neurons are always firing, and we can pick them up. That magnetic flux can be picked up. And here's how. The right-hand rule of magnetism says the following. Well, based on electromagnetism, just generally, anytime you have an electrical current, you will have a very small magnetic field outside that emanates around it. So the current, in this case, if you hold out your hand, like you see in the picture, with your thumb pointed up and your fingers curled, imagine that the current is your thumb. What you'll have then is a magnetic field that goes around your thumb in the direction of your fingers. Okay, this happens naturally. We don't have to create this. What that means then is if you have currents are propagating information in your brain, as we do with axons and dendrites, you will have small magnetic fields. And so just as another example, if you look to the right here, this current is in the blue arrows, following along the axon and the dendrites. If anytime you have propagation of information via the axon, you will get a very small magnetic field emanating around it. Well, we don't just have single axons. We have thousands of miles of axons in our head. It's densely packed with axons and neurons firing, and those, in essence, operating as currents. And if those currents are oriented in the right direction, then some of that signaling will emanate outside the head. You can detect it with these sensors. And so that's what MEG essentially does. Sensors all over the head, and you're picking up hotspots where there's more neural firing or changes in neural firing over time. And so the strengths of EEG and MEG are they have excellent temporal resolution. They're repeatable. There's no toxins. EEG is low cost. That's not true about MEG. And unfortunately, there's only maybe 15 to 20 MEG centers in North America. They're not as common as MRI. There's less spatial resolution. MEG is better than EEG in this regard, but it's not on the order of millimeters like MRI. And MEG is expensive, and again, there's fewer centers, and you can't see lower brain structures. Again, this is a signal that's just outside the head. That magnetic field needs to be emanating outside the head in order to detect it. So if you go deeper in the brain, you can't see it. There's one other technique before we get to MRI that I wanted to touch on. It's called near-infrared spectroscopy. And it's kind of a cool technique that really looks at how you shine light into the brain. And when there's more blood flow, it'll absorb that light. And so you shine light into the brain and then detect how much comes back. And so this is a blood flow based technique that really requires, it's based on the natural relationship between blood flow and neural firing. Where there's more neural firing, there'll be greater blood flow. And so you shine, there's near infrared light on the scalp, it diffuses through the cortex, there's brain activity and blood oxygenation changes, and then light reflected to the detectors is altered based on how much flow. It has pretty good temporal resolution against how well we can isolate events in time on the order of seconds. There's no radiation. It's relatively insensitive to movement, which is really important when we talk about MRI. It's portable, it's small, so you can take this to people's houses as opposed to being in a major medical center with a MEG or an MRI machine. Unlimited number and length of exposures. So it's, again, there's no risk to exposure, and it's low cost. And so with regard to movement, this is really important. You can study babies who often can't engage with some of the other techniques that we use because babies are going to move, right? And so that's really hard to do in an MRI machine, but you could use NIRS safely, FNIRS safely in infant studies. Some of the weaknesses, it requires much validation. It's been around a while, at least 20 years, and it still requires a lot of methodological validation to make sure that it's measuring what we think it's measuring. And it can be disrupted by non-uniform pigmentation. There may be differences if depending on pigmentation on its validity. Again, it needs much validation. It doesn't have the spatial resolution that MRI does. So the last technique that I'll discuss, and there's much more time spent on this in the following modules that when I'll talk about fMRI and then connectivity, is a blood oxygen level dependent functional MRI. And so this is the Siemens 3 Tesla magnet at University Park here in Penn State. And much like with the PET machine that you saw, you lay down on the table, and then the table goes back into the bore, the opening here, so that the brain function can be imaged. Unlike PET, there's no injection of a radioactive isotope. This is all non-invasive. So what is functional MRI? When neurons fire, there's an increased blood flow locally in order to replenish oxygen and glucose. And so fMRI is sensitive to changes in blood flow locally because of something called the bold signal, which I'll spend more time on in the next module. In essence, the bold signal is, as neurons fire, more blood flows into an area. And as oxygen is metabolized, that signaling changes between the deoxyhemoglobin, or the blood flow that has less oxygen, and the oxyhemoglobin, the blood flow that has more oxygen. And that ratio changes the signal that we're going to get across the brain. And so we can track where there's more neuronal firing and less neuronal firing based on blood flow. And again, I'll spend more time on that in the next module. This bold signal can be examined during tasks and at rest. And so some of the images you'll see from bold fMRI studies are these images of human brains, or structural images T1, T2, with the function overlaid, with the red typically meaning more neuronal firing and the blue typically meaning less, or a deactivation where there's neuronal firing drops below baseline. Again, something else we'll talk about more. What I want you to understand about bold is that it has reasonably high temporal resolution on the order of 1 to 2 seconds, and it has good spatial resolution on the order of millimeters because it's MRI. It does require the MRI machine, which is highly sensitive to motion, and so people need to be very still while they're engaged in different types of tasks. And the task-based fMRI we'll talk about in the next module as well. So some learning objectives from this module. Functional imaging provides opportunities to examine dynamic or changing cerebral processes. Most methods are non-invasive. PET does require injection, but NIRS does not, EEG does not, MEG does not, and fMRI does not. Those are all completely non-invasive. There's a natural trade-off between temporal and spatial resolution. As you saw with EEG and MEG, very high temporal resolution on the order of milliseconds, but you give up at knowing exactly where things happen in the brain. Spatial resolution, ideally you use MRI, which has really good spatial resolution, but the temporal resolution is not on the order of milliseconds like in EEG and MEG. fMRI, the temporal resolution is about 1 to 2 seconds. Again, the highest temporal resolution are in EEG and MEG. And the highest spatial resolution is offered by MRI approaches.